Dear students, we are going to see about the pure culture technique, which is a seed dilution and plating techniques, which is mainly used for the isolation of or enumeration of the bacteria either from the soil sample, gas sample and water sample. So what is pure culture? A pure culture is the population of cells that are arise from the single cell. Every cell in the culture is a genetically identifiable to the parent. That is called as a pure culture technique. In nature, the microorganisms that are exist in mixed population. It may be present in soil, water, human, gut. That's why the microorganisms are ubiquitous in nature. We can say it as a microorganisms are ubiquitous in nature to study the specific characteristics. Either it is for pathogenicity and antibiotic resistance. We must isolate the purest form of microorganisms. So Robert Koch, he developed the first method for obtaining the pure culture using the solid media. The principal isolation of this method is the main aim is to separate individual cells on the solid medium so that we can multiply them or we can distinct them or we can easily visible them. That is called as colony. We can easily visible the colony. Before that, you have to know about the CFU, that is nothing but colony forming unit. We can use the term instead of CFU, that is the bacteria, because a colony which arises from the single cell or a clump of cells like streptococci chains, one CFU is equal to one colony. So we are going to measure the colony forming unit after the isolation of this bacteria by performing the pure culture technique. Before that, we are going to serially dilute the concentrations of the available microorganisms by using this serial dilution technique. So a typical sample, either it may be a solid sample or liquid sample, water, soil, anything, the sample which contains billions of bacteria per ml of that sample. So plating directly the sample, which results in the clear complete lawn, and we can't isolate a single colony using that sample. So we have to perform serial dilution, which is the stepwise reducing the concentration of the microbial population. We are going to dilute the sample systematically. Usually we can perform the tenfold dilution to reduce the population to a countable range. The objective is to maintain the plates with 30 to 300 colonies. That is the standard number of colonies that can be measured. That is the logarithmic dilution of 1:10 or 1:100 we can use. So the procedure, we have to take 5 tubes or 10 tubes with 9 ml of sterile distilled water or saline water. We are going to add 1 ml of the sample to the first tube, and then this is the 1:10 tenfold dilution, and we have to, after mixing the first tube, we have to transfer 1 ml from the second tube. That's how we are going to serially diluted all the samples by diluting the concentrations of the microbial population. We have to repeat this till we are going to reach the 10th plate, 10th tube or 5th tube. And then we can use the spread plate method, pour plate method and streak plate method. So these three methods are the pure culture technique isolation methods. And we are going to, if we are going for spread plate, means we are going to spread the samples all over the already sterilized and solidified the agar plates. If we go for pour plate, means samples are mixed with the molten agar previously, and then we have to pour directly to the empty sterilized plates. That's a pour plate method. And streak plate method means it is a qualitative method for the isolation, and it uses the friction, that is inoculation loop. By using inoculation loop, we can use this streak plate method. So this is the separate procedures. So we can compare the spread plate method and pour plate method. In spread plate method, we are going to use the 0.1 ml of the sample, but in pour plate method, we can use 1 ml of the sample. Here the spread plate, we are going to use the surface only, but in pour plate, we can use either surface and subsurface. As we are already mixed the sample with the media, so we can use the surface and subsurface area. And in spread plate method, no heat is used, and it is good for the heat sensitive bacteria, but in pour plate there is a risk for the heat shock. And in oxygen, that is for aerobes, they are very well grown in spread plate method, and in the micro aerosolic bacteria, they prefer only the pour plate method. So this is the streak plate method. There are different types of streak plating: T streaking, continuous streaking and radiant streaking. In T streaking, we are going to divide the plate into 3 zones, and by using the inoculation loop, we are going to perform all the 3 zones separately, one after the another, after the incineration of the inoculation loop. In continuous streaking, we are continuously, without taking the hand, we are continuously dragging the microbial population from the top to bottom of the plate by using the zigzag movement and without taking the hand. This is the best for samples with lower bacterial loads. And radiant streaking, it uses the edges to the center. So we can often use it for fungi and specific colony types. So this is the formula for calculating the CFU. As I already know that CFU is nothing but colony forming unit. So number of colonies, that is divided by dilution factor into volume plated. Here if we have 30 to 300 colonies means we can statistically validate that points. If we are having less than 30 means then we have to say it as a too few to count. We can't count that number of colonies. Less than 30 means that is a high error. If it is more than 300 colonies means that is nutrient competition is already there. So it is too numerous to count. We can't take it into account for the calculation of CFU. Thank you, students.